Hello. Today, I'm going to be introducing our paper, An Approach to Learning the Hierarchical Organization of the Frontal Lobe, co-authored with the help of Duncan Cleveland, Charles B. Michael, Seema Mofakam, Yuri B. Salman, and Petar M. Jurich, being presented to the EUSIPCO 2023 conference in Helsinki, Finland. In this work, our primary focus is trying to understand and discover the anatomical structure of the frontal lobe, and in particular, the prefrontal cortex. The prefrontal cortex is a brain region which is vital for cognitive control, which we define to be our ability to adapt behavior based on our goals and context. It is believed that the prefrontal cortex is organized in a hierarchy of interacting regions within the prefrontal cortex that differentially contribute to cognitive control. However, despite much progress in recent years, the exact structure of this hierarchy is unclear, and many aspects of this structure are still being actively researched. In our current work, we consider the interaction of two regions within the PFC, the anterior cingulate cortex, or ACC, and the dorsolateral prefrontal cortex, the DLPFC. In a typical experiment, we record the brain activity of a patient as they perform a particular task. The patients under study have intracranial electrodes inserted into their frontal lobe as part of prior clinical treatment for epilepsy. During each trial of the experiment, the patients will perform the same task, which is composed of 3 steps. During the first step, the patient will be shown an abstract cue, which consists of a symbol that indicates whether they should pay attention to the shape or orientation of the object to be shown in the next step. In step 2, they'll be shown a particular object, which will have a shape, which is one of 3 categories, either oval, bow tie, or rectangle, and a particular orientation, which could be north, southeast, or southwest. Based off of what they observed in the first step, they should pay attention to either the orientation or the shape of the object being shown in the second step. In accordance with what they were instructed in the first step to pay attention to in the second step, in the third step, the patient must report what they have observed, either the orientation of the object or the shape, which they report using a saccade of their eye movements. To explain how a brain signal evolves in time, we resort to the autoregressive modeling paradigm. In this setting, the value of the signal y of t is represented as a nonlinear function f of the previous values of the signal, y of t minus 1 through t minus q, as well as the values of a neighboring signal, x of t minus 1 through t minus q, which are supposed to influence the brain region represented by the signal y. Any additional effects are represented by a noise signal wt, which explains what is not predicted by the autoregressive component. For shorthand notation, we'll use boldface y of t and x of t to represent the time window from t minus 1 to t minus q, where the model order parameter q is assumed to be fixed for now. To learn the nonlinear function f, we resort to Gaussian process regression. In this setting, we place a Gaussian process prior over the function, which means that for any finite set of samples that we have, the joint probability distribution for those samples is a multivariate normal. To make predictions using this model, we assume that there is a new point, y star, which we would like to predict with a corresponding input x star. Now, if we have training data of the form of previous samples, y1 through yn, we can use the probability distribution to condition out on the data that we have observed. Because Gaussian distributions are very nice to work with, this leads to a predictive mean and variance, which can be represented in closed form in terms of the covariance function of the Gaussian process and the previously observed samples. To estimate the function f, we just imagine the predictive mean of y star as a function of x star. In doing this, we're able to create a predictive model that, given a set of training data, is able to extrapolate to new data points that are previously unseen. In this figure on the bottom, I demonstrate this process for a recorded brain signal, where during the initial 800 milliseconds, we train a Gaussian process model based off of this technique, and in the following 200 milliseconds, we make predictions which are explained by the predictive mean and variance of the Gaussian process posterior predictive distribution. Gaussian process regression allows us to estimate the function f, but we still require a way to understand which input dimensions to the function, that is, which coordinate x1 through xd, most strongly influence the predicted output. To this end, we invoke the recently introduced concept of differential causal effect, or DCE for short, which takes the derivative of the function with respect to the xith coordinate as a measure of the sensitivity of the output to perturbations in the input. In this way, it is a measure of the local causal sensitivity of the output due to a change in the input. Since we're using Gaussian process regression, we're going to need to be able to take the derivative of a Gaussian process in order to measure the differential causal effect. But it turns out that this is a pretty straightforward procedure. If the function f is distributed according to a Gaussian process with mean function m and kernel function k, where m and k are assumed to be smooth or at least sufficiently differentiable, then almost surely any function that we sample from this Gaussian process is also going to be differentiable. In particular, the derivatives of this function are going to be distributed according to another Gaussian process, where the mean of this Gaussian process is mi, and the kernel is some ki, and these new functions can be determined directly from the original mean and covariance functions. Based on the previous slide, we know that to compute the expected value of the differential causal effect, we just need to take the derivative of the expected value of the original function. But if we remember, the estimate of the function was just the expected value of the Gaussian process anyways. So the expected value of the differential causal effect is just the derivative of the function that we already estimated. In this way, we can just use the previously learned function from GPR, and we observe that it can be written in this convenient form of a sum of the kernel function evaluated at different input points xn and weighted by some scalars alpha sub n. And we just need to take the derivatives of this thing. So what we get is a sum of derivatives dk dxi weighted by the alpha n's. So now I want to take everything that we've just learned about the differential causal effect and Gaussian process regression, and we want to use it to say something interesting about the prefrontal cortex. To this end, we're going to define the average differential causal effect as this sort of summation over the absolute value of estimates of the DCE, because this is an approximation of the average value of the DCE. And what this is going to tell us is that it's going to tell us the average causal strength of a particular lag of the signal x on the next value of the signal y. Now, we can further improve this estimate by averaging over every input lag of the signal x in this model to produce what we call the double average DCE, or the DADCE for short. Now, we will imagine this double average DCE as a function of the model order parameter q, and as we change q, we want to observe how the perceived causal strength of the signal x changes as we increase or decrease the window size. When we apply the double average DCE to the brain signals, we can already start to see interesting patterns arise. In the case of the ECC and DLPFC, we find that the double average DCE peaks for values of Q that correspond to a time window of around 7 milliseconds. What this indicates is that the most useful lags of the signal from the ACC for predicting the signal in the DLPFC have roughly a lag of 7 milliseconds, which corresponds to a biologically feasible propagation delay for a signal coming from the ACC and getting transmitted to the DLPFC. In addition to averaging over trials to estimate the propagation delay from the ACC to the DLPFC, we can also consider the trial-to-trial variation in this estimate. What we observe is that for different trials, there is an initial period in which the perceived causal strength is low for small values of the model order parameter Q. But as that model order parameter Q is increased, we see an uptick after Q passes some threshold. And while this threshold is trial dependent, it typically occurs between 4 and 8 milliseconds. If we vary the pair of electrodes that we are studying using the double average DCE, we see clear patterns arise, which indicate that the potential to detect a signal transmission from the ACC to the DLPFC has some level of location specificity. In comparison to other common methods, such as cross-correlation and pairwise Granger causality, we find that the double average DCE is unique in its ability to uncover these patterns, although future work is required to scrutinize these patterns and to determine their anatomical significance. As a first pass, though, this is a promising result because the double average DCE is a nonlinear method because it relies on the nonlinear modeling paradigm of Gaussian process regression, but cross-correlation and Granger causality, at least as implemented, rely on only linear models of the signals. The current work provides evidence that there is a causal influence from the ACC to the DLPFC. However, future work is needed to further scrutinize and verify the accuracy of this claim. In particular, if we have any hope of producing clinically relevant research using this method, we need to answer a number of important questions. Firstly, we need to address the robustness of the method, understanding how parameters such as the sampling rate will affect this measurement. Next, we also need to consider the problem of confounders or multivariate signals, in which 3 or more signals could be jointly acting together, and we want to be able to ascertain whether the ACC is directly influencing the DLPFC, or perhaps that perceived influence is originating or mediated by a third region, which we have not considered in the current pairwise analysis. Finally, we also want to consider the problem of time-varying causal strength within a trial, because as a patient is performing their cognitive control task, their brain state will change, and as a result, the causal strength between brain regions can also change. I hope that you found this brief introduction to our paper to be interesting. If you would like to learn more about our work, please read the full paper on the EUSIPCO website. Thank you for your attention.